Hello, everyone, and welcome to today's webinar, qPCR Research Testing for Pharmacogenomics, Workflow Validation and Content Selection. I am Kaylee Bach of Labroots, and I'll be your moderator for today's event. Today's educational web seminar is presented by Labroots and brought to you by Thermo Fisher Scientific. To learn more, visit www.thermofisher.com. We encourage you to participate today by submitting any questions you may have during the presentation. To do so, simply type them into the Ask a Question box at the left of your screen and click Submit. We'll answer as many questions as we have time for at the end of the presentation. You may also use the Ask a Question box to let us know if you're having any trouble seeing or hearing the presentation. And with that, I'd like to introduce to you our speaker for today's webinar, Victoria Quiett, Senior Technical Project Manager at Thermo Fisher Scientific. For a complete biography on our speaker, please visit the Biography tab. Welcome, Victoria. You may now begin your presentation.
Hello, thank you for the introduction. I'm happy to be here today discussing pharmacogenomics with you all. Again, my name is Victoria Quiett, and I am a Senior Technical Project Manager for qPCR applications. In my role, I help laboratories across the globe validate their qPCR genotyping and gene expression applications. For our agenda today, first we're going to be discussing a brief overview of PGX. Then we're going to move into our PGX portfolio and instrumentation, followed by specific details of that workflow. Then we'll move into what the validation process looks like for qPCR PGX applications and how Thermo Fisher can assist with that. Last, we're going to speak about content selection and customer experiences. All right, let's dive in. If you guys have heard about pharmacogenomics, you've probably heard it referred to as personalized medicine. This comes from the ability to assess a patient's genetic information and help determine how a patient can metabolize certain prescription medications. With this information, laboratories are able to provide guidance on the right drug for a patient at the right dose prior to beginning treatment, hence the term personalized medicine. When you have some type of health ailment or condition requiring a prescription med, the last thing any one of us wants to do is go back and forth with our physician trying to find a medication that works great for them. Pharmacogenomic testing takes out that trial and error method so many of us go through when we're trying to treat our conditions. PGX testing can contribute to multiple health fields, from cardiovascular, psychotropic, pain medications, and even cancer treatments. When it comes to the complexity of patient populations, you can see here a visual breakdown of the various groups a patient can fall into. For some individuals, drugs can work as intended and are beneficial and non-toxic to the patient. However, in other scenarios, patients can have the drugs become toxic and not beneficial. To further drive home the point on the necessity of PGX testing, you can see here on this slide that medications are just not one size fits all. Here on the left-hand side, a common heartburn medication is only effective in 1 in 24 patients. Overall, 90% of top-selling medications are only able to treat as little as 30 to 50% of patients. Not to mention the potential for adverse drug reactions. ADRs have been steadily increasing since the early 2000s. Along with this increase, so has serious ADR cases and even death. By the time it's clear that a drug just isn't working for a patient, their health has most likely further deteriorated. There is roughly $500 billion a year spent on the cost of prescription drug-related mortality from non-optimized medications. Adverse drug-related cases contribute to over 3 million physician visits a year, millions of emergency visits and hospitalizations, with over 100,000 deaths a year. Let's talk about how this can happen. The concentration of a drug in the bloodstream is going to rise and fall over time, based on a number of factors like absorption, distribution, metabolism, and excretion. In order for the drug to work for a patient, it must reach an effective concentration for a sufficient period of time. In some populations, we have what we consider a high copy number of a particular gene. So for example, CYP2D6. It's responsible for about 25% of prescription drugs. It relates to codeine. Most people have the standard 2 copies of this gene. However, in some cases, they can have 3 or 4 copies of a gene. In this scenario, we call them rapid metabolizers. This is where people with a high drug metabolism rate may excrete the drug so fast, they're not reaching that effective dosage long enough. Therefore, they're not getting any benefits of that drug. When a patient is on the opposite side of this scenario and only has 1 copy of a gene, they are slow metabolizers, where the drug is just going to sit in their system and could reach toxic levels within the bloodstream. The content for PGX testing with SNP or genotyping assays are targeting what we call DMEs, or drug metabolizing enzymes. Every organism, including humans, have a protective enzyme that modifies certain types of potentially toxic foreign molecules. The modifications make the molecule easier to excrete and will likely reduce toxicity. Some drugs, like codeine, are inactive until they are converted to an active form by DMEs. Rapid metabolizers risk producing toxic concentrations of that active drug in their bloodstream. So to recap on the impact PGX testing has in our community, by identifying how patients respond and don't respond to medications, hopefully before the drug is administered, can not only save billions of dollars a year, but also thousands of lives. Now let's move in to how Thermo Fisher can assist with this type of testing. We have a couple of different options for you to choose from. Our largest solution can run thousands of different targets and is a complete solution for genotyping, copy number variation, and HLA analysis. We have more smaller targeted panels that are more clinically actionable that can be executed on our Ion Gene Studio and AmpliSeq panel, which can also be beneficial with new variant discovery. Our most common solution, though, is the far right. Our customers love the Applied Biosystem QuantStudio 12K Flex. The reason for this is the low cost per sample and the fast turnaround time this instrument has to offer. Here's a clear breakdown of each one of those instruments and their capacity with how long it takes to get through the full workflow to an end actionable result. You can see here in red on the far right, our OpenArray technology specific to our QuantStudio 12K solution is the quickest, but our microarray solution on the far left in blue can support more targets, but has a longer turnaround time. Now let's move into what the workflow looks like for our most popular solution, the QuantStudio 12K. Our QuantStudio 12K can support various formats to help with different numbers of samples a laboratory can receive within a given day. Our OpenArray technology on the left here was designed for higher volumes in mind and can handle more targets than on the right-hand side our 384-well Tachman array plate. This is geared towards lower numbers of sample volumes in a given day, but can run on more instrument types like the QuantStudio 5, 6, 7, and 12K. For our OpenArray technology, it comes in the shape of a rectangle and it's on the left-hand side here. This one chip has a total of 3,072 through holes or reaction wells. Within each square or subarray is 64 total through holes. There is a total of 48 available subarrays, and each one is dedicated to a given sample. The sample throughput will change based on the number of targets selected in a given panel. It's going to vary from customer to customer, and we can offer multiple formats to fit your desired custom selection. In the right corner, you can see how the various formats break down, and you can change the number of targets you are interested in relative to how many patients you expect to run in a single day. As a reminder, this is only one OpenArray chip, and you can run 4 OpenArray chips in one single QuantStudio 12K run. We have 2 panels for our QuantStudio 12K OpenArray solution that is not custom and is considered what we like to call off-the-shelf or inventoried panels. They come in format 60, which holds a total of 48 samples, but has 60 different PGX targets on this panel. Then on format 120, can hold 24 samples, but it'll have 120 PGX targets. We also have our inventory panel for a 3-4 well format. It can hold a total of 6 samples per plate. Some of the benefits of selecting our made-to-stock inventory products, one is that the turnaround time is much quicker and they can show up to your laboratory within a matter of days, where a custom format could take anywhere from 6 to 12 weeks, depending on the targets that are selected. So proper planning and inventory must be done by the laboratory to prevent any gaps in their workflow materials. The pricing is also cheaper for our inventory panels. I think another good benefit to our made-to-stock content is the extensive research our R&D team has done to ensure that the selected targets in the panel are comprehensive and relevant to the current market. The workflow for our OpenArray product is fairly simple and utilizes 3 different types of instrumentation to assist in the workflow. The first part of this workflow is your samples are going to go through your standard accessioning and intake process for your desired sample type. The sample type options for this test can be buccal swab, saliva, or blood. Once you're ready to extract your samples, you can utilize our Kingfisher Flex that can extract 96 samples at one time using our multi-sample ultra extraction kit and protocol. This part of the workflow can take around 2 hours to complete. Then before we load it into our AccuFlow robot, we want to quantify our DNA samples to better understand the concentration we're putting onto the qPCR technology. This is very important because not only is it going to optimize the detection of your results, but it's also going to help save you money by not running samples that are too low of concentration to yield quality results. After you quantify your samples, you can load them onto your AccuFlow robot and ultimately the PCR for your genotyping experiments. The OpenArray genotyping experiment is going to take around 2.5 hours to complete, and the CNV experiment is going to take about 1.5 hours, totaling about 4 hours of runtime. Lastly, you're ready to analyze that data, which can take 30 minutes to an hour, depending on your experience. For our copy number variation experiment, we have multiple assays to choose from. We have 3 different assay options targeting CYP2D6 specific to exon 9 and intron 2 and 6 for the hybrid alleles. When we run these CNV experiments, we're going to run them in 4 technical replicates to help with the accuracy when we're analyzing our results. You're going to analyze these data points in a separate software called CopyCaller software that's going to help you assist in calculating the appropriate copy number for your intended target. The last step of the workflow is translating that data that was previously analyzed. Typically, most of our customers utilize a third-party service to help generate an actionable report for a physician. There are 2 really great companies that can assist with this. One is Coriell Life Science, and the other is Translational Software. Once the process is set up with either company, it's going to be very simple for the laboratory. The lab technician is just going to drop the exported file that was previously analyzed into a particular folder on their desktop. This folder is connected to your LIS software and your third-party company that you've selected to translate the data. They will then pick up the data from a HL7 or API data transmission, and within minutes, you're going to have a beautifully generated report that a physician can take action with. Now let's discuss the analytical validation services for PGX testing. As many of you know, prior to beginning your new LDT test, you're going to have to go through a rigorous validation study adhering to your specific accrediting body. You can see here all the variations in these definitions about different organizations regarding a validation, and they're going to vary, but the end goal is the same, which is for the laboratory to establish with evidence that their test method delivers reliable results through the intended application. Every organization is going to have very specific requirements, and it's important for you to reach out to your specific accrediting body, whoever you subscribe to, CAP, CLIA, New York State, and find out what they specifically require for their validation studies. Knowing this information prior to spending any money or effort on your validation until you have a clear understanding of what is required, it's absolutely critical for the success of your laboratory. It's going to be very frustrating for a laboratory to think that they finished a validation, but have to go back and complete additional work to get approval from their accrediting body. Some labs may feel comfortable taking on a validation on their own. However, there's a lot of factors that might not be considered when a lab makes this decision to take it on themselves. We know customers who do make this choice, and it could take around 6 to 8 months for a laboratory to get up and running. This is really because the amount of work that's truly involved in a validation process. A lot of time goes into the project planning. You can see here the first 3 months are dedicated to project planning alone. Then executing your AV plan and collecting all that data and analyzing it and working on that necessary documentation to meet the needs of your accrediting body could take another 3 months. This is why if you let Thermo Fisher assist in your validation, you're guaranteed to finish the validation work 75% faster. Let's talk about some common struggles laboratories experience when executing a validation on their own. I think the first one here is project management. There's a lot for a laboratory manager to do on their daily responsibilities within the lab outside of designing a validation plan. And when it comes to designing your plan, first you have to decide your panel content, decide your instrumentation. You need to code your LIS software. You need to work on the reporting side. You're going to have to order all of your consumables relevant to the protocol. You're going to have to write those protocols. And then finding the time to do all that, relative to what your day-to-day responsibilities are, I think it's difficult for some of our laboratory managers to manage all of that. Another common struggle is that control material. It's going to be really difficult to find full coverage on the control material for your panel where all 3 potential genotype calls for every assay are represented on the panel. Not only is this complex to find, but you're most likely going to have to resort to a custom synthetic plasmid to meet that need. And when you're designing a custom plasmid, it comes with a fairly high cost. So once you source all the material you need, you have to design that plan to meet your accrediting guidelines. And I think the next step that comes pretty difficult for our customers is actually analyzing all that data. Most of the time, they don't expect how much data is going to be generated from an AV plan. It's very common for us to analyze around 30 plates per validation customer. So if you're new to analyzing data, it becomes very challenging going through with a fine-tooth comb, looking at all that genotyping data. Another pain point customers don't think about is compiling all that data and putting it into a presentable form that's going to meet your accrediting standards. As you can see here, how much time and effort it takes to undergo a validation, outsourcing all that hard work and stress is a great way to save time and money. Letting Thermo Fisher assist in your analytical validation is going to help you get to that finish line a lot faster. So let's kind of talk about what that process looks like for our customers who go through a validation with us. First, we start with the selection of your panel equipment. Once your equipment and your consumables have arrived for the lab, that needs to be installed and calibrated by our field service engineers. They will also give a tutorial on the instrumentation. A project manager like myself will be selected for your engagement, and they're going to be working in the background while you're getting your consumables and your instruments to your laboratory. During this time, they're going to be selecting and designing that control material needed for full panel coverage. They're going to assist in that workflow optimization and write the laboratory protocol templates specific to the workflow. Then they're going to design that analytical validation plan and documentation outlining the scope of the experiments. We're going to provide on-site support from our field application scientists that will execute a 3-day workflow training in person prior to the validation. And this is to help ensure that the lab techs are prepared and they know how to execute every step of the workflow before we begin our validation experiments. This is just going to help us yield optimal data points for the AV. Then we're going to give the same FAS scientist that did your training visit again for 3 more days to execute your validation plan. A project manager will take about a week or 2 to analyze all of those data points and form a final summary report for your accrediting body. So here's a short list of what you're going to be provided if you purchase our complete package for AV support. You're going to be provided, like I said, a dedicated project manager. You're going to have a dedicated field application scientist for training and analytical validation. We're going to provide all the protocol templates and do any workflow optimization that's needed. We're going to include all of the control material needed for the validation. So for genotyping, we use a mixture of Coriell human cell line controls as well as synthetic plasmid controls. And this is going to give us full panel coverage for every potential genotype call within every assay. In addition to that, we're going to select 5 real human donor samples of your choosing. This can be your lab technicians or other staff members that are going to undergo the full workflow. So you'll collect that buccal swab or the blood or saliva sample, take it through extraction, all the way to the end qPCR. Those same samples that were selected, the 5 human donor samples, are going to be sent out for next generation sequencing so we can have an orthogonal comparison to your qPCR. Then we're going to provide all that data analysis in the final summary report. We also have a new 1-year health check that is really similar to a 1-year warranty, and this comes automatically with the purchase of our complete AV package. If there's any change to your workflow, like perhaps you wanted to update an assay or change the extraction kit, or maybe you just got a software upgrade, we're going to support you in a revalidation or bridge study to incorporate your new change into your previous validation work. And that's good for 1 full year after your AV is complete. Last on our agenda today, we're going to start talking about real-world customer experiences and content selection. When selecting your panel content for PGX, the first step is really understanding who you're going to be marketing this test to. Perhaps you already have multiple physician connections from other types of testing going on in your laboratory. Understanding your target market and who's going to purchase this test from you is key to designing the optimal panel. Some laboratories prefer to have smaller, more targeted panels specific to each discipline, like you see here. Our inventoried panels that I mentioned earlier can be considered more of a larger, comprehensive panel, including pain, psych, and cardiotropic drugs. However, these can still be used for the desired smaller panels that are ordered by a physician and to help meet medical necessity. And you can do this by masking other drugs that are not relevant to the practicing physician. This can easily be done through your LIS software. We have numerous published success stories on TaqMan genotyping assays, as well as our OpenArray technology. However, there's one that I want to highlight that is a peer-reviewed publication co-authored by Coriell Life Science and Thermo Fisher. It focuses on the utility of pharmacogenomics, as well as the technology, user acceptance, and implementation, but most importantly, that economic impact. In this paper, it's concluded that all of these factors are really at the tipping point, and the best time to take advantage of PGX testing is actually right now. And now, from the public experience dealing with COVID, it really helped increase awareness to molecular testing in general, and I think has, in general, helped increase in that common citizen and raise that common standard of care that they expect from their physicians and laboratories. So let's conclude. The benefits of choosing Thermo Fisher for your PGX testing solution is really our expertise in this industry and our success with our technology for really over 10 plus years. We can get your lab implemented faster with this technology by our customer support paths and training and validation services. We offer numerous options to choose from on our instrumentation to assay content selection. Let Thermo Fisher handle your pharmacogenomic testing needs in the future. Now, I would like to segue into our Q&A portion of the presentation.
All right. Well, thank you so much, Victoria, for your informative presentation. As you did just mention, we will now start the live Q&A portion of the webinar. So to our audience, if you have a question that you'd like to ask, please do so now. Just type your question into that Ask a Question box and click Submit. We'll answer as many of your questions as we have time for. All right, so let's get started. We've already got some great questions coming in from the audience. So Victoria, this first question asks, what concentration ranges are expected for the test?
Thank you, Kaylee. That's a great question. So it's going to vary on your format. So for the open array technology that we have, we recommend 25 nanograms per microliter as the recommended concentration. But for our 3D4 TACMan array pre-spotted plates, we're going to recommend 5 nanograms per microliter. During our analytical validation services, we're going to span a full dynamic range, starting at that recommended concentration for a given format, and then work our way down so the customer can see the full range down below the manufacturer LOD. Thank you.
Awesome, thank you. Another question I have for you here. What is the most common sample type?
I think the most common sample types our customers prefer is the buccal cheek swabs. There are others like blood or saliva that can be utilized, but I think customers prefer the buccal swab because they're easier to transport, they're easier to collect, but there is some downfalls when using buccal swabs. One would be that variability that you can get across patient to patient. Not every patient or collector is going to follow that recommended protocol on best practices for buccal swabs. So feel free to check out our website and you can see a document on best practices for buccal swab collection. And you can also reach out to our field application scientists for more information.
Great, thank you. All right, another question here. Does a laboratory have to use a third-party company to translate the genotyping data into an actionable report?
That is a good question. No, they do not have to utilize a third party. It does come with a cost using a third party software to help generate that report. But we do have a solution at Thermo Fisher. You can use our allele typer software, though I think a lot of customers do prefer to use that third party outsourcing program with Coriell or Translational because it's just a quicker process and it adds some time using the allele typer software to analyze again. So I think customers prefer outsourcing.
Great. And again, we're getting some great questions in from our audience and we have time for a few more, so we'll continue. Next question asks, what is the recommended extraction kit that can be used to process multiple sample types?
Yes, our multi-sample ultra version 2 is the best for multiple sample types like the buccal swabs, blood, and saliva with minimal changes to that protocol to accommodate the sample types.
Great. Next question, what can be used to quantify the extracted samples?
There's a lot of options that can be used for the quantification method. Our preferred method is what we call RNA-seq quantification. This can be done on your same QuantStudio instrument you are running your genotyping or CNV targets on. It can process hundreds of samples at one time, which I think that to me is the appeal. But there are other methods like qBit or the nanodrop, but the input is a little bit different. RNA-seq is going to be more accurate and can handle more samples, hundreds of samples at a time.
Great. All right, next question here. Does the same AV consulting services apply with Genexus or a QuantStudio 7? Do they offer the same assays?
It does offer the same assays. You can select any content you would like for your given instrument. And the analytical validation services still apply for qPCR or next generation sequencing or any type of platform you're looking to establish this on. We can give you support. Great question.
Yes. All right. Well, thank you so much, Victoria. This does conclude our session for today. So before we go, Victoria, I want to ask, do you have any final comments for our audience?
I really appreciate everyone jumping on here to learn more about pharmacogenomic and our testing solutions here at Thermo Fisher. We're happy to get you going with our analytical validation services, so feel free to connect with your local sales reps for more information if you're interested.
Wonderful. Yeah. Well, thank you again, Victoria, for your time today and your important research. Before we go, I do want to bring to your attention the survey at the bottom of your screen. If you would like a quote to discuss pricing for Thermo Fisher PGX Research Solutions, please select yes and submit, and a team member will be reaching out to you shortly. So I would also like to thank Labroots and our sponsor, Thermo Fisher Scientific, for underwriting today's educational webcast. And I'd like to thank the audience for joining us today and for their interesting questions. Questions we did not have time for today and those submitted during the on-demand period will be addressed by our speaker via the contact information you provided at the time of registration. This webcast can be viewed on demand, and Labroots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. So until next time, take care, everyone. Goodbye.